Greetings to the viewers. In this session, we will learn about the packaging of DNA. Before we learn, we should know that why is packaging of DNA required? Why do we need to pack the DNA? And if there is a need, then how can we pack the DNA? How does packaging of DNA takes place? So let's go back and first find out that why is packaging of DNA required? So let me tell you that if we take the example of the human genome, right? The one set of human genome, if we talk about, one set of human genome means 23 chromosomes. So in 23 chromosomes, the length of the DNA which is there is approximately 2.2 meters. Right? How do we get this length of DNA? Because we know that in the human genome, there are 3 billion base pairs. We have found out this, right? And also we know that the distance between the two base pairs is 0.34 angstroms. So thus we have calculated that in one set of human genome, the approximate length of the DNA is 2.2 meters. Now, the question is that how such a long DNA gets fitted into the nucleus of the cell. The cell itself is a tiny structure, and then the nucleus inside it is even smaller. How does it happen? It seems as if I have given you this much of wool and I say that you have to fit it into this much box. How will you do it? What will run in your mind? Definitely you will start thinking of rolling and folding in such an effective way so that it gets fitted into the matchbox, right? So something like this also happens in the cell. So we'll see that how is DNA packed into the cell. For this, I would like to tell you that there was a scientist named as Woodcock who observed and reported that there are certain thickenings in the chromatin fibers. He observed that. There was a scientist named as Audet. He termed these thickenings, he called these thickenings as nucleosomes. He also observed, and he said that these thickenings are nothing but nucleosomes. He also said that these nucleosomes are actually made up of DNA and certain proteins which are called as histone proteins, right? And when these chromatin fibers, they condense at the time of cell division, right? We know that the chromatin fibers, which are found in the nucleus, they condense into chromosomes at the time of the cell division, right? So when they form the chromosomes, they are condensed and thickened structures, then these thickenings are even more visible, right? And these thickenings, as Audet says, that these thickenings are nucleosomes and they are made up of DNA and the proteins called as the histone proteins. So let's see the detailed structure of the nucleosome. So if we see that a nucleosome is made up of histone proteins, these histone proteins are positively charged proteins, which are made up of the basic amino acids lysine and arginine. These are the two main amino acids, basic amino acids that will make up the histone proteins. And these histone proteins are of various types. Say, for example, there is H2A, H2B, H3, and H4. These four basic types of histone proteins are there, which are present two in number. Each protein is present twice. So, like H3 is there, H3 is there, H4 is there, H4 is there, H2A is there, H2A is there, H2B is there, H2B is there. Okay, so you see that H2A, H2A, H2B, H2B, then H3, H3, H4, and H4 also twice. So this type of an octamer is produced. You can see it here also. If we say that this is H2A, H2B, H3, H4, and the second copy of each is also present at the back. It will be visible, and so it forms the octamer. Now, around this octamer, the DNA is wrapped. Okay, how it is wrapped? You can see that it makes one complete turn and then 1/3/4 turn, and it moves out. So the length of the DNA, which is used up in wrapping the histone octamer, is around 146 base pairs. Or we can also say that the DNA takes 1/3/4 turn around the histone octamer, right? And this DNA, which wraps around the histone octamer, is actually called as the core DNA. And this structure is called as the nucleosome. Also, a point to be noted that when the DNA leaves the nucleosome, you can see this DNA. This DNA is called as the linker DNA, which is also a part of the nucleosome. Right? So you can imagine something like that, a big ball, which is made up of 8 small balls and held and wrapped around by a thread-like structure, which is DNA. Now, you can imagine that this structure will be a loose structure wherein the DNA or the thread-like structure can stripped off at any point of time, and the balls will fall apart. So we feel that there should be something, a clamp type of a thing, which can hold this structure together. So there comes the role of another protein which acts like a clamp to hold the nucleosome. And this protein, can you see here, is called as the H1 protein. But this is also a histone protein. But this protein will not become the part of the histone, nucleosome. This is a histone protein, H1 protein, which will help in holding the nucleosome together, but it will not be the part of the nucleosome. You can see here, the nucleosome basically actually consists of histone octamer, the DNA, which is inclusive of the core DNA and the linker DNA. And the H1 protein, which is acting like a clamp, it is not the part of the nucleosome. The reason is that it has been observed that this H1 protein is not inherited. Right? So this is how the packaging of DNA is described. And this model which has been described is called as the nucleosome solenoid model. The way it has been described is called as the nucleosome solenoid. What this model tells? It tells about the packaging of DNA inside the nucleus in the most effective way. Right? So let me also give you a picture. See here, one nucleosome, linker DNA, and then another nucleosome will come, and this will keep on repeating like this. So if we go back where Woodcock and Audit, they observed that there are certain thickenings in the chromatin fibers. These thickenings can be well explained with this model, right? And before I take you to the further folding and packaging of DNA, let me give you a 3D view to make you understand that how actually the model looks like. You see how the 8 balls of the histone proteins are there, wrapped around by the core DNA, right? It leaves the histone octamer and the linker DNA moves out. So this is how it appears. Now here you can see that the single DNA, right? The single DNA, then it is wrapped around the nucleosome, that is the histone octamers. The further folding also takes place. Further folding and looping takes place. It keeps on condensing. It keeps on condensing and folding till it takes the shape of a chromosome. right? So this is how the packaging of DNA is described. So this is all for today. Thank you.